Hello everyone. First, thanks for the introduction. This is Xinjian from Zhejiang University. My lab mainly focuses on the neural mechanism of motor control and mental disease. Today, I'll present one of my work which addresses a lot of difficulties during my research in motor control. The title is Deep Behavior Tracking, a novel behavior tracking method for laboratory animals based on deep learning. This work mainly focused by my student Guanglong Shen. As we know, behavior measurement and evaluation is one of the key methods to understand brain functions in neuroscience, especially with respect to the movement and social behaviors. To do this, different behavior paradigms have been developed and is used to evaluate animals' movement, anxiety, social behavior, disease development, and sleep order, and the medicines. As I had told before, our lab most focus on the motor control and social behavior. So, locomotion measurement is super important in my labs. In our labs, we also use this kind of behavior paradigms, include open feed, one maze, treadmill, and three-chamber test. In these conditions, the environment becomes more and more complex. And sometimes, the tracking in these conditions becomes super difficult. Also, when we want to study the neural mechanism of different behaviors, in our lab, we use optical simulation, calcium imaging, and the electrical physiology recording. During these conditions, the image line or the optical lines will enter the imaging, makes the image much more complex. And even sometimes, during this optical simulation, the light will come out and also makes it much more difficult tracking. We also use different animal models in our lab, include mice, rat, and another one is the non-human primate marmoset. The marmoset usually withhold in a metal grid box. In these conditions, the color of the background and the animals is similar, which makes it much more difficult to tracking. To give the importance of the behavior evaluation, several methods have been developed to tracking the animal models, include the straightforward method and the video tracking method. For the straightforward method, it will add some hardware on the animal's head or the body. It can't work in some conditions. This setup also effective only use some simple animal types or operate complexly. Another method is based on the video tracking. But the video tracking, they also have two methods. One is background subtraction, another one is the deep learning. For the background subtraction, there are commercial software have been developed, include Limelight, Animaze, Iteration, and TopScan. For the simple conditions and the clear little background conditions, this software can help the neuroscientists a lot to evaluate animals' behaviors. However, this kind of the method don't works on the complex conditions. Then rely on another method called deep learning. Recently, some machine learning algorithms showed high precision in object detection, such as deformable parts model and region convolution neural network and YOLO method. Based on the CNN, several toolbox based on the learning have been developed, include DeepBhvTracking, Leap, DeepBhvTracking, text, and dance. The broadly used is DeepBhvCart. In our labs, we also test the DeepBhvCart. For the DeepBhvCart, they will mark the different postures of animals for the training dataset. Then, during the tracking, they also can detect both the animal posture and the growth movement. However, this method is very slow, and both in tracking and the detection. This one also requires supercomputers, including the CPU and the GPU, and also training the animal models for many times. For examples, in our labs, we get detected in one condition, it can't use another condition. Also, we train the animals in one animal can't use for other animal in same condition. So what we're thinking, whether we can find some of the even simple method, because we don't want to know the locomotion, we don't want to the details of the movement, details of animals' posture. So what we're thinking is we use some simple deep learning algorithm and then combine with the background subtraction method. To do this, we choose YOLO. YOLO is also a deep learning algorithm based on CNN, and sometimes it has super fast speed and can even up to 155 frames per second. This method identifies targets by classification. It transforms the detection process to a regression problem, and predict the coordinates of targets of the bounding box and classify the probabilities. So this method can detect the target from the full imaging through a single network and make it easier to optimize for better performance. So for our tracking method, we have three parts. The first one is detector training, second one is YOLO detection, and the third one is spectral subtraction. For the detector training, we should label the animal position in the training and the validation data set. And then after training, we see a detector. Then we use the detection parts to get the bounding box of the animal's target position. Last, we use the background subtraction method to remove background in the bounding box and getting the shape animal and then getting the centroid animal. Below is the image flow for the tracking process. In the stage 4, 3, and 4, we getting the bounding box, and 6 and 5 and 6 we remove the background, and 7 we get the morphology or the position of the animals, and 8 we estimate the centroid of animal. In our conditions, we also test the different neural networks and the different pixel size. We found with the increase the pixel size, the training time increased. And also, this net shows the longest time for the training. We also checked the detecting precision and the speed, the relation between the precision and the speed. We found the RedNet 50 at the pixel size of 515 TeF has the highest speed, has the best performance. So we choose these parameters for all the conditions in our conditions. Here we compare the full method, the tracking for all the four methods. The first one background, YOLO only, DeepBhvCart, and the DeepBhvTracking. As we can say, in the background method, sometimes they also show the animals and also the lines. Sometimes when the hand intruder is here. And for the YOLO one, it also only gets the bounding box, and the bounding box, the size of bounding box can jump to different positions. For the typical cut, even we have the same training data set, and sometimes the training dot also can jump to other positions. For the behavior tracking method, we can clearly get a centroid or animal. Then we can see the tracking road for each method. We can say for the background subtraction and the deep lab cut, we can see a lot of jumping from one place to another place. This for the background subtraction because it detects a lot of background. For the deep lab cut, it's because when the hand put here, this method says the hand also be target. For YOLO and DeepBhvTracking one can achieve good performance. But for the YOLO detection one, they also can see small rectangle positions. That's because the jump of the size for the animals is jumping for different times. We also check this, says the tracking road in the chamber test. We can say the background subtracking is worse in the Gaussian conditions, and also for the YOLO typical lab cut and the typical behaviors can give the clear trace. However, after calculation, we found the DeepBhvTracking have decreased the training time compared with the deep lab cut. Also, DeepBhvTracking have the faster tracking speed than the deep lab cut, and they have relatively high precision than deep lab cut, YOLO, and the background. And also, we can get the move speed for all the animals. It is showed as lowest for the DeepBhvTracking than all the others. We can get the clear trace for the L-shaped maze and the L-shaped maze and the three-chamber maze. We want to know whether it works for other conditions. We check the treadmill, L-shaped maze, and three-chamber white mouse in the three-chamber, and also inverted V-shaped maze. We can tell it works all the conditions. What I want to say is in the treadmill, in these conditions, the room light is very dim and also black mouse. Both the other method can define the animals from background, but our method can define the animals from these conditions, can show the relative movement of the animals with the treadmill. Another one, we also checked with the marmoset in this metal grade box. We can say it can clearly get the running map of the animals of the marmoset. Another one is we usually want to look at the two animals in same condition, same environment. But we don't know our method works or not on this condition. Theoretically, this YOLO method should work on these conditions, but they have two white mouse is thus too similar that this method can differentiate with them well. What we can do, we add a blue marker, a different color marker on the animal's body. This method clearly can detect the two animals separately. We also get the trace for different animals. So this method is fit for different animals, different paradigms. Then we want to evaluate animal behaviors. First, we use the open feed to do this in different genetic animal models, include wild-type, PRT2, and F1-1. PRT2 is a movement disease model. F1-1 is emotion or the social development animal model. Clearly, and A is the running map of the animals. The B is timing map. Clearly, and spent more time on the corner than the center. C is the speed map. We can see the animals running faster in the center than in the corner. We compare the average speed, time, and average speed in three different animal models. We found FRM1 has higher speed than all the other two animals. This result also have been visible in others report. Last, let's make a summary for our result. First, we developed a method. It's called DeepBhvTracking, a novel behavior tracking method for laboratory animals based on deep learning. In this method, we combine both the usual and the background subtraction. Second, using DeepBhvTracking, moved animals can be tracked accurately in complex environment and can be used in different behavior patterns and different animal models. Therefore, DeepBhvTracking can be broadly used in the study of neuroscience, medicine, and machine learning algorithms. Last, our articles for our method is posted on GitHub. You can free download it and use yourself. And last, thanks Guanglong Shun for all the help to make this software, to this toolbox, for our labs to the behavior tracking. Last, thanks for your attention. Welcome to Hangzhou. And any questions are welcome.